our first mini talk series for our PHS and joint with DBE and clinical biostat seminars. So our two speakers are both local. They are junior faculties at Zhang and UW. So the first speaker is Dr. Zhang, who joined our DBE program in 2022, right, as an assistant professor. And his research interest is broadly in causal inference, design and observational studies, instrumental variables, and application of statistical tools to public health sciences. So today he will share two problems about what he found in clinical vaccine trial design, right?
Right.
Okay, welcome both. So each talk will be about 25 to 30 minutes, and we'll leave some time after each talk for questions.
Okay, great.
Yeah.
Yes. So our plan today is I will introduce like two mostly ongoing submitted-ish papers I've been working on. And I'm going to mostly just introduce the problems. You can find the details in the paper. And of course, we will have a little bit of time for discussion. Okay. So the first part is the title is the debiasing the hazard-based time-varying vaccine effectiveness estimates using vaccine-irrelevant infections. This is joint work with many colleagues. The leading author is Ethan Ashby. He is a PhD student at UW. Okay. So here is how we come up with this problem. So I started by looking at some of the large vaccine clinical trials that VID did during the pandemic. The most important one is this COVE trial. It's a phase 3 randomized double-blind placebo-controlled study of Moderna's COVID vaccine in the United States. And this trial actually leads to the approval of the Moderna vaccine and also publishing the New England Journal of Medicine in 2021. So one very interesting aspect of this trial, relatively less well known, is that in this trial, in addition to collecting these SARS-CoV-2 clinical endpoints, the trial used a multiplex assay. So when the patient has a symptom-triggered illness visit, they not only can detect SARS-CoV-2, but also 20 other quote-unquote off-target respiratory disease pathogens. These are all tested by this multiplex PCR platform. So I will refer to these as the acute respiratory illness. Off-target meaning they're not SARS-CoV-2. So a person thinks he was sick, he comes to an illness visit, give him a test, turns out not to be SARS-CoV-2. The person got influenza, RSV, blah, 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 etc. Okay, so here is a nice plot. So there are about 16 different pathogens that we collect during the trial. Of course, the majority of them, these are, the stacked bar plot. You see, the majority of them are SARS-CoV-2. These are SARS-CoV-2, but the different colors, the black, light blue, yellow, etc., they are other coronavirus or some other influenza, rhinovirus, etc., adenovirus that people collected during the clinical trial. Okay. X-axis is the timing of the trial, right? You are seeing the wave. So this is the different COVID waves. This is the booster part of the COVID. This is when the Omicron was circulating. But anyway, you always see some other viruses circulating in the background. And we collected these endpoints as well. So in this paper,
so when you say off-target, other than
SARS-CoV-2, yes. These are all others are SARS-CoV-2. Okay. So this is a figure that I took from a published paper that we published in the American Journal of Epidemiology. So here, a very natural question is, it is good to quote unquote verify, depending on perspective. So what is the SARS-CoV-2 vaccination, this Moderna mRNA vaccine, on these off-target endpoints? Okay, so this is during the blended phase of the trial. This is probably, because people have a lot of ideas about off-target endpoints, negative controls. This is a golden opportunity to actually quote-unquote verify whether or not this is a genuine negative control outcome. So here, these are the cumulative incidence curve of those off-target endpoints in the blinded phase of Moderna trial. And as you can see, not too surprising. Again, depending. Some people even think there might be an effect. Some people think, of course, there's no effect of SARS-CoV-2 vaccines on off-target endpoints. But anyway, this is data. There is no effect. Okay, at least that we can detect during the blinded phase of the COVE trial. So, and also we also do some additional analysis. So we also use some method developed by Dean Foreman. So here we are looking at this is the V waning estimate. So it's how the ratio changes over time during the blinded phase. This is against SARS-CoV-2. As you can see, pretty high throughout, very high, above 90% efficacy. And this is, you repeat the same analysis, but on these off-target endpoints, they're almost the incidence ratio is 1. So this is essentially saying those who are vaccinated, those who are vaccinated, but who remain in a risk stat versus those in the placebo arm at the risk stat, their hazard ratio of catching those off-target endpoints are essentially 1 throughout the trial. Yeah, combined. There's yeah. Technically, these mRNA vaccines should be very specific because they should bind to the very specific epitopes, etc. So we don't believe they are cross-reactive. Yes. Although practically speaking, I think the majority of the viruses are rhino, RSV. There are like 3 or 4 things. Although we list like 16, most of them are very sparse. Most of them are rhino, I believe, RSV, some influenza maybe. Okay, so this is all good. They are not too surprising. So somehow this thing doesn't work.
I think you just need to click.
Yeah. Okay, great. Yeah, these are all great. These are in a blinded phase of the trial. Okay, so in 2021, so we started to offer a booster, offer the SARS-CoV-2 offer booster to all the study participants. So it doesn't matter if you are a randomized to placebo or the primary series. After the placebo people cross over to the primary series, and then after a while, we offer booster to everyone. So any people who have received the primary series are eligible to receive a booster. And the same testing procedure was adopted. So again, this is the SARS-CoV-2 and off-target endpoints during the booster phase of the trial. And the booster phase, the Delta and Omicron were primarily circulating during the booster phase. So the interesting thing here is we want to estimate the booster efficacy, essentially. Booster, 3 dose versus 2 dose. The thing is the booster is not randomized, so the actual data is people come to receive the booster. Some people receive it earlier, some people receive it later. Ideally, initially, philosophically, we can imagine it as some kind of an individual level, a stepwise design. So, people are initially everyone was not boosted, and then some people receive booster. Among the remaining ones, at week 2, additional people receive booster, et cetera, et cetera. But again, the receiving booster is not randomized. You can do whatever you want, right? So again, in this AJE paper, what we did is we repeat the same analysis, okay? So we are looking at the vaccine efficacy. This is relative vaccine efficacy because it's 3 dose versus 2 dose against the Omicron and Delta separately. So this is a naive analysis, something that one would otherwise do, pretending that the receiving booster is sort of a quote-unquote randomized. Right? If you believe that's randomized, then it has a little bit closer to a causal interpretation. But when we repeat the analysis on these off-target endpoints, here the interesting thing is we have some evidence that it appears that people who receive the booster appear to have a higher incidence of catching those off-target endpoints, at least initially. Okay, so if you remember, if you look at the same plot during the blinded phase, it's very nicely 1. But here it deviates from 1. So what we think is at least this could be taken as some evidence that there is some bias, essentially. People who are in the risk stat, people who receive the booster but not infected versus who did not receive the booster, they're not necessarily comparable, essentially. So this is all it's telling us. So that's the end of that AJE paper. And the question is, we want to sort of, at least to some extent, account for these unmeasured, probably time-varying frailty process. So we believe there is a frailty that change over time that we didn't fully capture. Okay? So here is a simple model that motivates the framework. Although that thing was developed in more generic terms, here is a simple data generation process that satisfies those abstract model formulation, and that's easy to say. So a simple model is there's 2 hazard models, one for the primary endpoint, one for those off-target endpoint. For those off-target endpoint, there's a baseline hazard and there is a U of t that is a frailty process. So it's a measured frailty that might change over time. And here, this is the primary endpoint hazard function. Again, it has its own baseline. They share the same unmeasured confounding process, which is an assumption you need in order to identify things. And there is, this is the usual thing. This F function is the vaccine waning function. So this is V is when you receive a booster shot, T is the current time. So the difference is time since your booster. So this is essentially capturing how the V is waning over time. So this is that's essentially it. That's all the motivation. So we saw these things. We won't have a way to account for that. And this is a simple model that help us account for the essentially those off-target time to event endpoints in when we analyze the vaccine efficacy. Okay. Yes. We can stare at it for a while. And I would say the strongest assumption is clearly, you can throw in some baseline covariates. I did not include them, just for simplicity. You can throw in some baseline covariates. The strongest assumption is this sharing U of t. You can, of course, argue it is U of t here, but maybe a slightly different process affecting the off-target endpoints. But yes, in order to borrow some information, this is what we need to make this type of assumption. In causal inference, we give it a name as some kind of equiconfounding type of assumption. Essentially, this is some kind of equiconfounding. But the U of t is left unspecified, just like the baseline hazards. And our goal is just to estimate this function f in the presence of these nuisance. Yes, I put the reference here. So there are details of how we actually make inference for it. Yes, and you can efficiently estimate f while leaving U unspecified, and also baseline non-parametrically modeled the pure hazard-based formulation, not causal inference. They not necessarily are causal. You need to impose additional assumptions so that this function f in this statistical parameter has a causal interpretation. But the parameter is the same. This second part is more like just fulfilling our curiosity. When is that parameter has a causal interpretation? That's everything. You don't have to believe any of these assumptions. You can always estimate and treat it as a statistical parameter. It's fine. We use hazard-based all the time. Or the main objection is the hazard. There's some built-in selection bias because over time, people in the risk set are increasingly not comparable, right?
What is the bias?
Yeah, this is the emitted variable bias. There's a, yeah, please.
So the U of t essentially, you get information of U of t based on the correlation between the non-target infection
and the primary endpoint.
Shared.
Exactly. And actually, the thing is slightly trickier. I don't have time to get into all the details because even in the randomized trials, people write papers on the depletion of susceptibles bias, which is actually in this paper we actually can claim, so that is a way of sort of also capturing a little bit of depletion of susceptibles. Because the idea of this depletion of susceptibles is that in these two arms, over time, people remaining in the risk set became increasingly not comparable, right? That is reflected. If that's the truth, then their hazard of catching those off-target endpoints should gradually deviate from 1 because they are increasingly not comparable. Their underlying frailty becomes more and more different. This is not what we actually see in this real data, in this data set. So actually, we even make a point in this paper that we saw very minimal depletion of susceptibles bias in this. And this is also a way, even for randomized trial, you can use it to sort of detect. And maybe, detection does not require any additional assumption. Correction requires additional assumption. So the bottom line is you can do this. And you can say there is minimal evidence of depletion of susceptibles, which is not too surprising because the instance rate is not very high. So they're mostly the entire randomized cohort, right, over time. So yeah, anyway, I think for the sake of time, we need to move on. Yeah. So that's sort of, there are a lot more details, but that's sort of the idea. We have a time to event negative control outcome. we want to take account for that, essentially. And slight generalization is in some trials, we don't have money to do those multiplex testing. What we do, sort of a poor man's version, is we use those test negatives. So there are people who come to us, test SARS-CoV-2, the person turned out to be negative. You can sort of treat it also as a, as a negative control. This is not the most ideal, just because you can say this is a false negative. There could be some suppression in the viral load, blah, blah, blah. It's more comfortable if the person actually tested positive for something. But you can, but that's a poor man's version, which is okay, I guess. Yeah, if you pair that with some additional symptoms, it's probably okay. Okay. Yeah. Well, actually, this if you go back to real data, so yeah, this is the booster phase. This is for the Delta wave. This is for the Omicron wave. Let's look at the Omicron wave. If you ignore those off-target endpoints, just do the quote-unquote naive analysis. This is the VE waning curve that you would derive, and these are the bias sort of quote-unquote bias corrected curve. You see, they are a little bit higher after we correct for those off-target endpoints, which is again intuitive, just because here we are saying people who receive the booster have a little bit higher tendency to catch those off-target endpoints. We believe that's probably because if you receive the booster, you just are less cautious, for instance. It's more likely for you to be exposed to viruses. So if you take that into account, a naive analysis might underestimate the booster efficacy. Of course, it's not what I expected initially, but it is what it is. So initially, we thought we might overestimate, but it is what it is. Okay. Yes.
Can I ask a question? Can you go to the previous slide? No, the one that you showed. Sorry. Yeah, this one. So for the non-targeted, before the booster, what's the curve look like up to the whatever?
Yeah, before the booster, it is essentially look like this.
Oh, okay.
So if you look at but this is 2 dose versus no dose. But then we gave them a booster. So this is 3 dose versus 2 dose.
So then somehow it gets 1 and then jumped and the booster happened when the booster happens?
Yeah, this is a good point. So technically the ending point of this analysis is not strictly overlap. It's not strictly contingent to the other, because there's also a crossover period, and also because the people who randomized to placebo, we gave them the primary series vaccine. So, and after a little bit, after everyone received the primary series, we then offer the booster, and then they voluntarily take the booster. Does that make sense? Yeah. So these are not contingent. There may be something that happens between, but this is but anyway. So yeah, let's move on to the second part here. So yeah, the second project is, I think, this is a joint work with Peter and Oliver. So this work is actually, I think, is probably more relevant to folks here. So we study, the goal is to, again, estimate the cumulative incidence or the cause-specific cumulative incidence. We want to estimate this for I use updated vaccine regimen. You can think of it as an improved immunotherapy or whatever. It's just improved treatment, okay? So we, possibly different study population. So the data we have a historical trial. We have because this is very natural in our setting. We do have those historical trials of an old Moderna vaccine, okay? And then they update the regimen. We do not run a new trial. Also, ideally, we should run a new trial. The FDA probably want us to run a new trial. It's sometimes very time-consuming. What we actually do is they do a so-called immunobridging study. So they compare the new vaccine to the old vaccine on the study population, our target population. Instead of recruiting like 10,000 people and record our clinical endpoint, we recruit 200 people, only record the immune markers, and that's it. And if the new vaccine has a higher immune marker, this is typically taken as evidence that the new vaccine is better than the old, and they typically approve the update of the vaccine. Okay, so the data structure is a clinical trial that measure everything: clinical endpoint, biomarker, baseline cohorts, and the data from immunogenicity study, essentially. So which does not capture clinical endpoint. So what we want to do is we want to ask this counterfactual hypothetical question: what happens if this immunogenicity trial essentially just keep going, enroll 10,000 people, let them develop the clinical endpoint? What will be the cumulative incidence? Okay. So yeah, unless there's some literature here, I have to skip them. for the sake of time. So the data structure is baseline covariate, treatment, biomarker, mediator. Depends on your point of view. I think of this as a mediator. Okay? So this is a mediator of the treatment's effect on the outcome. And the T is the time to event. Of course, in reality, possibly subject to sensory. And we do have another immunobridging data set, DB, baseline covariate, treatment, mediator. That's it. So here the treatment is 1, 0, old vaccine, placebo. This is the new vaccine and the old vaccine. That's it. Okay. So yeah, this is just to differentiate data from which data set or which population, how you think of it. So I'll just describe to you the estimate here. The estimate, what we want to estimate. So in this new trial setting for the population, the condition X, but the X is marginalized over. Okay, what's the time to event? If I just give everyone either 1 or 1 prime, either the old vaccine or the new vaccine. That's it. So I just want to understand the cumulative incidence curve. Okay. Yes, the interpretation, cumulative incidence at time t naught, had the immunobridging study population receive the approved vaccine or the new investigational vaccine, or the old treatment and new treatment, essentially. And of course, you can also study the relative risk. Yeah, I'm going to skip the tasks. So essentially, the task is just to approve those updated vaccine regimen. This is our primary goal, using just the immune marker or the surrogate endpoints, however you want to think of them. So yes, I will tell you there are 2 key assumptions. Yeah, so in code, these are all code assumptions, identification assumptions. The way I think of it is, of course, these are not ground truths. So I lay them out because you need to know what assumptions one needs to make in order to answer the question I posed. Okay, you can also relax them if you want, but these are, in some sense, the minimal assumption you need to make in order to ask the question that I just posed. There are 2 assumptions. The first assumption is, if you intervene, give people the same vaccine, and set the immune marker to the same level for the same people, then the cumulative incidence or the expected outcome of some transformation of time to event is the same in these 2 trial settings, which is, of course, not going to be always true. They can be conducting different time periods, blah, blah, blah. But this is something you need to make in order to answer that question. In reality, you can relax a little bit. You can normalize by the placebo incidence, for instance, in order to make it a bit more likely to hold. But this is the first key assumption. The second key assumption, which is also the most important, and this thing does have a very biological interpretation as well. So this is essentially saying there's no relative controlled direct effects when you compare the old vaccine and the new vaccine. This is not saying the vaccine has no direct effect on the outcome. It's saying these 2 vaccines, relatively speaking, they do not have direct effects. So each of them can have direct effects, but they need to be the same in order for you to generalize or transport from 1 to 1 prime, which is, again, pretty intuitive. Yes, so this is the formulation of this assumption. And these 2 qualities are, again, you just treat them as leading cases, and they form the basis of whatever sensitivity analysis you want to relax this assumption, blah, blah, blah. And so this is the primary result is under these assumptions, we can identify the target parameter. This is the efficient influence function, which then we construct a semi-parametric efficient estimator based on this EIF using cross-fitting. It has those usual robustness properties. So it is consistent and asymptotically normal under the union of these 3 models. So if you correctly specify these 2 models, it's correct. These 2 models is correct. These 5 models is also correct, essentially. And also in the paper, we also do everything but for the cause-specific events. So we do allow, different endpoints. Peter does not like the competing risks just because technically they're not absorbing, but you know what I mean. Though they have multiple clinical endpoints. Okay, so we can also estimate cause-specific events, which is useful because you can have multiple serotypes or multiple pathogens that co-circulate, right? So you want to estimate cause-specific. So Omicron-specific cumulative incidence, Delta-specific cumulative incidence when they co-circulate. But again, I don't have time to Good. I do wish to, yes, I can use the last 3 minutes to just show you the actual the data. So this is a trial that we run, a booster trial. Enrolled 300 people, randomized to, in one stage, randomized to different Pfizer-BioNTech vaccines. And later in stage 4, enrolled 200 more, they randomized to BA1, these two bivalent vaccines, the Omicron-containing vaccines. Okay, so we can answer several different questions. For instance, the first question we can ask is, so in stage 2, they did not receive these Omicron-containing vaccines. I'm actually curious, so what happens if we gave those stage 2 people those Omicron-containing vaccines? What would be their cumulative incidence? Okay, so it's probably easier to look at this way. So here is the data. We pretend we have their immunogenicity of for instance, of the stage 2 Omicron-containing vaccines and the stage 4 BA.4/5 containing vaccines. You see the stage 4 BA.4/5 containing vaccine has a higher biomarker. It is more it elicits more immunogenicity. Okay. And I also observe what is the cumulative incidence curve corresponding to these people. I want to ask, if I have this curve, I have this box plot, if I further give you this box plot, can you construct me a cumulative incidence curve using our methods? And this is the curve that we construct using the methods, essentially, right? Also not surprising, a little bit more immunogenicity, a little bit lower incidence. That's essentially it. So this is the type of question we can ask. And another use case is, yes, this is probably more of a scientific interest. So we always make this assumption, no control direct effects. I want to assess this assumption among the stage 2 participants, because in stage 2, I have those prototype vaccine. Prototype vaccine just means use the original Wuhan Hu inserts, those D614G, the old inserts, and Omicron-containing vaccine. So here, this is old vaccine, this prototype vaccine, this is Omicron-containing vaccine. This is their cumulative incidence curve. We want to assess the assumption of no control direct effects. Okay, so one way we can do that is, under that assumption, we actually estimate what would be the cumulative incidence curve for these Omicron-containing vaccine recipients. This is what we get. So I get this curve using this blue curve plus their box plots, essentially. Okay, and the reality is I actually observe what is the actual Omicron-containing incidence curve. I compare what I estimate under that key assumption with the actual data. In reality, you don't have this. In other applications, but here we do have this curve, so we just compare them. You can compare their cumulative incidence, or I just compare the difference at day 188. This is the 6 months endpoint, and the difference is clearly excludes 0. So this provides evidence that the assumption under which I estimate this curve is wrong. That's it. So we can reject this assumption. So there is controlled effects. So those Omicron-containing vaccine, prototype-containing vaccine, again, very intuitively, you see, they only differ a little bit in immunogenicity with this biomarker. The cumulative incidence curve differ by so much. Clearly, there are some biomarker that there's some other pathways that mediates this effect. Very simple. That's essentially the intuition. And we're just trying to formalize this using the mathematical language. Okay, I think that's it. We do have a paper. Yes, it's mostly done. It will be archived hopefully by the end of the month. That's it. Thank you.